to video 2 in the genetics video series. Today's video topic is going to be about mRNA transcription. Now, a few things before we get started. You should know that before we really go into today's lesson, you should have completed a quiz on DNA basics. You also should have read and answered the questions on the transcription and translation code that was the reading about building a castle. So those are those things you want to take care of before you watch today's video so that you can follow along with us a little bit better. And now, as you are following along with today's video, you should be reading mRNA transcription, the process. So this is a reading that's on the back of the transcription and translation overview. Now here's just a quick review of what we've covered so far. So the molecule you're looking at right now is known as DNA, deoxyribonucleic acid. So we can see a couple of things here in our DNA. We see the individual bases here, here, here. We also see those same bases in kind of a fun 3D form along here. Alright, so now remember our bases are adenine A, thymine T, guanine G, and cytosine C. Now, each of our bases is then connected to a backbone, and then what we get is double-stranded DNA molecule. Remember, the purpose of the other strand is to make sure that the letter order doesn't get messed up, right? It's called a complementary strand. So now, as we take a look at DNA for a second, one thing that we have to realize is how important DNA is to the body. So the purpose of DNA is that it provides information to every protein in the body. Now, this is important because proteins play a role in pretty much everything we do. So proteins play a role in how fast your toenails grow to what color your eyes are. So pretty much everything you are physically is a result of your DNA in some way, shape, or form. And the DNA, the way it controls you, is it makes proteins that allow your body to function. Okay, we'll learn a little bit more about proteins. But there's a pretty big problem when it comes to DNA encoding proteins, and it has to do with the little information in our cell unit. So if you remember from the cell unit, DNA is inside of the nucleus. But if you have a good memory, you'll remember that the organelle that's responsible for building proteins is actually called the ribosome. Now the issue comes in that the DNA is inside the nucleus, but the ribosomes are not. So somehow the information has to get from the DNA to the ribosome. Now you may think this is simple as, okay, well if the nucleus just lets the DNA go, the DNA will just kind of drift out of the nucleus and then will go and make its way over to the ribosome and problem solved. But there's an issue with that. The issue is that the nucleus is actually protecting the DNA. You remember, the order of letters is incredibly fragile. It's very easy to change. And if you were to change something like that, all of a sudden your code becomes very different. One little letter makes a huge difference. So the cell's job is actually and not allow it to get changed. The way it does that is by trapping it in this protective nucleus here. Right, but the problem comes in that somehow this information still needs to get from here to here. All right, how do we do that? Well, that's where transcription comes in. All right, what the cell does is it actually reads the DNA inside of the nucleus. It makes a copy. That copy is what you saw here. That copy is known as mRNA. The mRNA is then able to freely leave the nucleus, make its way over to the ribosome. The ribosome reads the mRNA and is able to make the proteins. Alright, so what we're going to be talking about today is that process of copying DNA into mRNA so that we can have information get sent out of the nucleus to the proteins. Right? That's called transcription. How does it work? Well, in order to understand how it works, we have to take a quick review of our story about building a castle. You remember from our story about building a castle, when we turn DNA into RNA, that's a process known as transcription. turning RNA into an amino acid sequence later as proteins is something called transformation. We read a little bit about translation, but we'll get a whole video on it. It's actually a topic of this video. Now, where does transcription happen? Transcription happens inside the nucleus itself, whereas translation happens outside the ribosomes. These are two major processes that are going on that we're going to follow. Now, if you remember back to the last video lesson, we learned about how to make complementary DNA. So essentially what you're doing is you're making the two strands of DNA. If you know what's on one strand, you can actually tell what's on the other strands. And the reason you can do that is because DNA is pretty consistent in that whenever you have an adenine molecule A, it always pairs with a thymine molecule C. Thymine, in turn, always pairs with adenine. And if you were ever to see a cytosine on one side of the DNA, there's always a guanine on the other side. Whereas if you were to see a guanine molecule on one side, cytosine is always on the other side. And that's how you make complementary DNA strands. Now, this is a little bit different for DNA to mRNA. For one thing, the copy is only a single strand of information. It's actually only a small chunk of the DNA. Remember, DNA is about 6 billion base layers long. Right? Most proteins are not that long. Most proteins are only a few thousand letters. So what happens is that the DNA knows which section of it codes for individual proteins. All right, those sections are known as genes. So one gene will code for one protein. So what will happen in transcription is that only one small gene out of a whole sequence will get copied into mRNA. You don't want to make a copy of the whole DNA at a time. That would get so long. All right, and this way you're only copying one part of the gene that you need. Now, again, this looks very similar to making complementary DNA, except for one small difference. In mRNA, the molecule thymine doesn't exist. All right, so everything else works exactly the same. That thymine on DNA still pairs with adenine on RNA. Cytosine on DNA would make guanine on RNA. Guanine on DNA would make a cytosine on the RNA. But the difference has to do with this one right here. Since thymine doesn't exist in RNA, adenine instead pairs with this new nucleic acid known as uracil. Uracil. All right, we abbreviate that U. And that's one of the big ways that you can tell the difference between DNA and RNA is whether or not there's uracil. So now let's practice doing this. This is our original DNA sequence that was found inside of the model that we showed before. So this is the DNA sequence. We would start with the RNA. The RNA copy of this, well, if you remember, adenine here would pair with uracil. Thymine would pair with adenine. Guanine with cytosine. Cytosine with guanine. Thymine with adenine. Adenine with uracil. Guanine with cytosine. Guanine with cytosine. And cytosine with guanine. And so what we get is this whole chunk of DNA, I'm sorry, mRNA, that's into a little bitty copy. So what that looks like is a chunk of mRNA. Now let's get some practice doing this individually. So go through this right now and see if you can pair these on your own. All right, this is a longer DNA sequence. So remember, the first step is to split it up into 3. So we put some little spaces. And then if you were to make an mRNA copy of this, it would look like the T, the thymine would pair with an adenine, cytosine with guanine, guanine with cytosine. All right, then now if we look at this one, the thymine would pair with adenine, guanine with cytosine, cytosine with guanine. Now if we look at this one, guanine with thymine, guanine with cytosine, thymine with adenine, adenine with uracil. If we look at this one, adenine with uracil, adenine with uracil, thymine with adenine. This one, adenine with uracil, cytosine with guanine, guanine with cytosine. We can keep going on and on until we finish our mRNA sequence. Now you can get some practice on your own. Take a look at the mRNA transcription process. And at the end, there's some questions. Make sure you answer those questions before you take your quiz. Once you answer those questions, you should be able to be successful on that quiz.